and that was KD. And he was the first one to describe it, and this is the reference to his paper. And what I want to do is describe a little bit about what is happening. I won't be able to explain to you why it's happening, because the answer to that is we don't really know, and well, that doesn't stop us from trying, but we don't really know. But what's happening, and if you look at the title of the paper, two keywords are muscular atrophy. And you'll find out that scientists really love jargon, primarily because they don't want people who are not scientists to understand what they're talking about, even if they're talking about something that's pretty simple. So they could have said muscles getting smaller or muscular shrinkage, but instead they invented a new word, atrophy. So all it means your muscles get smaller. And so we can, you go on the internet, look up muscular atrophy. This is one of the first pictures you can steal, and it has a picture. Well, maybe steal isn't the correct word, borrow. I'll put it back. So we have, here's a normal biceps, and here's a biceps that's a lot smaller, and that's atrophy. A smaller muscle doesn't work as well as not as strong. Now, that's what's happening now. So our muscles are atrophying, they're getting small. Now, why do muscles atrophy? Well, there are probably a number of reasons, but the one I'm going to try to talk about deals with its relationship of the muscles with the neurons that control them. Muscles are very dumb. The only thing they can do is contract, and they can't do it on their own. They have to be told when to contract. And who tells them to contract are neurons, special nerve cells called motor neurons. And motor neurons have a nice structure that we can generalize, and they have basically two major parts that we're going to care about. They have a thing called a cell body, which has all the parts that cells have, and that's usually hidden somewhere up in the spinal cord or in the brain. We're going to see spinal cord here. And then they have a process, a long elongation called an axon that leaves the spinal cord and then travels to wherever the muscle is. So some of these can be pretty long because they have to go from your spinal cord to your big toe, for example. And these axons then, when they get near the muscle, branch out and terminate on individual muscle cells. Each muscle cell is a tube. That's this thing right here. So this has a picture of probably about 15 muscle cells in there. That would be an extremely tiny muscle. Your muscles easily have thousands of fibers, or can have thousands of fibers. But each muscle cell only receives stimulation from one motor neuron. So, for example, this green one, this motor neuron, terminates on a number of different cells, but each of these cells only receives from the green one. And they all work as a team. So there's no way, if the green one gets activated and sends a signal, all of the muscles that it's connected to are going to contract all the cells. And that's the way we control the strength of our muscles, is by controlling the number of neurons that are activated. Now, you can actually see this close up. Here's a real picture, and this is a muscle fiber, and this is one axon that's branching, and you can see how it terminates, onto the neurons. And you say, oh, that's pretty cool. Well, if you were to go in with a pair of scissors and cut the axon, well, that doesn't normally happen. It's not necessarily a very nice thing, but if you do that, two things can happen. Either it can kind of regenerate, which doesn't happen all the time, or the muscle cells now have nobody to tell them to contract. And if they have nobody to tell them how to contract, they don't contract because they're not that smart. And if they never contract, what happens is they shrink. And the reverse is opposite. If you use your muscles get bigger. Well, if you don't use them, they get smaller. If you've ever worn a cast for a long period of time when you're immobile, you would know that, say a leg, your leg would be much smaller because the muscles have atrophied. So one way you can have a muscle atrophy is to lose the motor neuron. Now, this is an example of that experiment I just said where they cut a motor neuron. These are muscle cells, and this time they're cut in cross sections, so they're really coming out at you. Big long tubes. And these guys down here look nice and big. These guys up here look nice and big, but in the middle you can see they're a lot smaller. These were the only ones that have lost their motor neuron. These were not cut, these were not cut. And you can see, there's a drastic change in size. And again, the bigger it is, the more stronger it is. So what does that have to do with KD? Well, if we look in the spinal cord, and we look for motor neurons, first of all, this is the typical sort of spinal cord. I'm sure you really have no idea what you're looking at other than this strange shape. possibly. Well, the spinal cord can be divided very grossly into two major parts. The white matter, which kind of looks white, and gray matter, that's kind of looks gray. They're going to say, no, wait a minute, I thought they always gave special terms. Well, this was probably done by a grad student. It's not making remember that. The white matter we don't care about. That doesn't have any cell bodies. That just has fibers that are moving up and down. And that's the outer part, the part here that's kind of grainy. The inner part, and not, people always seem to call this, I think it's another name, but it looks kind of like a butterfly. Just the black part, not the gray part on the outside. This is the gray matter. And it's in the gray matter where you find nerve cells, the cell bodies of nerve cells. And at the very bottom of them, these regions down here, which have special terms called the anterior horn, but that's not a big deal. All you have to remember is that they're on the bottom. That's where the motor neurons exist. So if we look at a real slide of a stained spinal cord, now it's not really easy to see. What the white matter is very clear. That's all this stuff here. You can see the outline of the butterfly. Yes? Jameson, can you see it?
Yes.
And you can see these big purple things in this lower half. Those are motor neurons. So they're huge. They're probably about the biggest.
Now, what are the other nerves that bring the feedback back to your brain? Are those
These are the fibers that are going up and down, so like the telephone cables going up and down. And the cells that are in here are kind of involved in sending, there's also cells up here that send them up.
And we're losing those too.
We are losing those, but we don't notice it as much.
Oh.
So we are losing more than just the motor neurons. I mean, I shouldn't say that. The bottom of my feet are completely numb. And it's now growing up my leg.
Yeah,
So
that's what I noticed. If I'm standing up, I'm feeling like I'm falling unless I just touch something.
Yeah.
And that feedback...
trying to stand on one foot.
Yeah, right.
Again, you're losing your sensory and the whole of reflexes that keep you doing it. And that's why I think we talked at one point on some place about reflexes and... Okay, so we are, but I think, so while we are losing them, somehow we never care about that. And when I say we, I mean kind of the whole community. We're really more worried about the lack of muscle movement. So, this is a normal, am I in the way, by the way? If I'm in the way, you have to tell me. You're going to tell me. You can see the big cells, and these are
Motor neurons.
very good. And here's one up close and personal. I mean, it's pretty big. It doesn't look like much, and it's saying, But if you look at a spinal cord of someone who had KD, this was taken from one of Katsuno's papers from a long time ago. He was here last year. You may or not remember. He received a KDA grant about 2 years ago, I think. And this is half the spinal cord. And it's also stained in different ways, so it looks different. The white matter is dark, and the gray matter is light in this one. And I know it's there simply to confuse people. But the point is, here is that anterior horn. This is where you should see lots of big cells, and what do you see there? Not very many. And so it appears that we are losing, that's with KD, and we're losing our motor neurons. What does that do to the muscles? That makes them atrophy. So it's thought, that's the main model, is that what's happening is we're losing our motor neurons, causing the muscles to atrophy. Is that correct? Maybe. There's always a possibility that the loss of motor neurons is secondary, that something else is happening that's causing the muscles to atrophy on their own, and that it's not related to this. We don't know. But this is the model that has been followed by probably most of the researchers, but it's not the only model, so I just want to stress that. Now, the second part of that original paper talked about a sex-linked recessive trait. First of all, it's probably wrong. It's probably not recessive, but that's not important to us. I'm not even going to talk about the genetics. Elise Schindler is here from NIH. If you have questions, you can ask her. She's fantastic. She's very nice. But when you want to cure something or to solve a disease, the first thing you have to know is why you're having it. So you can attack it the correct way. So if you know, for example, you have a bacterial infection, you know you attack it by killing the bacteria. You take antibiotics. If you have a viral infection, you can take antiviral drugs, or you can get immunized. You know, and those are ways of combating that. Well, this, because it's a genetic disease, because it's linked, that tells us something that isn't necessarily obvious to everyone. This tells us that the problem with the disease deals with a protein that's not functioning the way it should. And that'll be more that'll come more clear. As I almost said more clearer. I really wanted to say that. Wanted to be more clear as I progress, as we keep going. But that's important. So now we know that because it's genetic, we don't know why we know that, but we know there's some protein that's messed up. Now, when we say protein, I forgot to say about the laws of heredity. Anyhow, that's no. This is what you think of when you see protein. Unless, of course, you're vegetarian, then you look at it in this way. This is not what we mean by protein. These are, the reason we even eat proteins is so we can take the proteins, break them down, and use the parts to build our own proteins. The proteins we eat, we always break down. What we're talking about is a protein that our cells make. And so I'd like to talk a little bit about proteins. If you don't think about them as meat and tofu, what do you think of them as? You can say washing machines? Well, I would be a little more general. I'd say it's a machine. Or you can think of it as a tractor, or any other machine you can think of. Proteins do things in the cells. And anybody know who Alton Brown is? The proteins are the ultimate unitaskers. They do one thing and they do one thing really well. Okay. And sometimes they don't even do something, they just sit around and look good. It could be a structural component. No. So if you want to think of your proteins in your cells as machines, things that have certain functions, and each one has their own special function. But that's not quite what we have here. And when you have proteins, they're made of, it turns out, one thing that's nice is they're all made chemically out of similar components. So it's as if you're making them not out of all the different things you make machines, but you're making them out of Legos. Because there's only a certain set of Lego shapes and colors, you're limited by that, and they fit together in certain ways. However, with Legos, what can you build? You can build a washing machine, you can build a tractor, you can even build the Statue of Liberty. So with proteins, when you build them, you can make amazing wide variety of machines. Now, the building blocks of them, and they're a little bit different than Legos, are things called amino acids. I'd like to tell you that right now this will be in the Jeopardy, but you don't have to know. There's 20 different amino acids. I put up the structures to make it look like it's real chemistry. It's really not important to us what they look like, just that there's 20 different ones. They have slightly different properties from each other, so it's kind of like letters of an alphabet. There's 26 different letters in an alphabet, and what can you do with them? You can spell an infinite variety of words by doing what? Varying the sequence of the letters. Same way we can build different proteins by varying the sequence of amino acids. Unlike Legos, proteins are actually, that's right, to be correct on telling this, that it's really a chain. You can only build really in one dimension. Okay. And you just keep adding them to the end. So actually, instead of looking like Legos, they look kind of more like this. But the nice thing, it's like a snake, though. Then it can wrap around and make different shapes. And it turns out the shape that a protein makes, just like in any other machine, is critical to its working. If it doesn't fold the correct way, it doesn't work. And so here is a really bad example, of a protein that has just been synthesized at the top, and it goes through a series of stages until it forms its final shape. So it's, even though you don't see the amino acids, the string represents the chain of amino acids. For whatever this protein is to work, it has to be, let's say, in this final shape. All these others, and so why does it fold that way? And the answer is, well, it's made to fold that way for the most part. They do it somewhat naturally. And that's based upon the sequence of amino acids. And so they fold into that particular shape, they're able to work. So the shape, how it folds in 3 dimensions is very important. So that's all that matters. That's what I'm trying to get across, is that it has to fold the correct way. And just to show you there's a wide variety, these are different, and the colors of course make it a little cute. These are an example of 12 different types of proteins. And some of them are very complex, like way down at the bottom, where they have maybe 15 or 20 different chains that come together, but they all have a specific structure and that structure is required for its function. If you change the structure, it doesn't work. So how do you change the structure? Well, one way is to change the temperature. If you heat up a protein, that changes the three-dimensional structure of the protein and no longer works. If a protein no longer works, guess what happens to the person in them if it's an important protein?
Dies.
They die. So what do you want to avoid so your proteins don't fall apart like that?
Getting stuck in a refrigerator.
Well, we don't want to get too hot. Right? That's why if your temperature goes above, and we're going to use Fahrenheit because most of us are from the US. I feel so embarrassed. I'm about 110 degrees. That's enough to cause enough of your proteins to change so that eventually you're going to die very quickly. Another thing we avoid are extreme acids or extreme bases. Another one is you have to have the right concentration of water and the right concentration of electrolytes. If you run a marathon, silly thing to do. You lose a lot of water, your electrolyte balance goes too high. That can cause proteins to misfold, cause them not to work, and you die. So I mean, the point is some of them are, some of the reasons you die and you try to avoid, it's all because of protein change. Now, what in the world does that have to do with KD? Before we do that, let's review, because we are having basically a quiz afterwards. So proteins are doers of the cells, and they do things. They're made of long linear chains of amino acids. Sounds like a Jeopardy answer. They fold into three-dimensional shapes determined by the sequence of amino acids. And if it misfolds, it is dysfunctional. That's a good word.
I like that.
I know. It's kind of scientific because I could say it doesn't work.
Yeah.
And it might have serious consequences for the cell depending on whether the protein's important. One of the questions you have, you might have a couple, but one of them is, well, how do you know what the sequence, or how do your cells know what the sequence of amino acids are? And basically, this is our quote day. Genes are important because they are the blueprints for proteins. We heard that yesterday. Lenore had that exact word about blueprints. And then you can read the rest. We don't care about that. So it turns out, the way that you know what the sequence of amino acids is in a protein is what your genes tell us. So when you inherit something from your parents, the ability to say have blue eyes. The reason you have blue eyes is because you have genes that make a protein that somehow, some way, cause you to have blue eyes. It's all because of proteins. Now, that means now we have to look at the DNA. DNA is another long, giant molecule. It's even bigger than most proteins. Huge. But it's only made of 4 separate types of molecules. They're called nucleotides. So unlike amino acids that have 20 to give a sequence, for DNA there's only 4. That's what genes are made of. And they abbreviate them with letters. They're U, they're G, they're C, and they're A. Now, if you know about DNA, you might say, what about T? But we're not going to worry about that. So just assume there's 4 letters. Somehow, when you have a sequence of nucleotides, so here's a sequence, UUU, GUC, AA, UGA, and mind you, DNAs are easily millions of bases long, or nucleotides long. So it actually continues all the way down until you hit Florida, and all the way down until you hit Arizona. So I mean, it's a pretty long molecule. This is just one small section. Well, let's imagine that this is a section that codes for a particular protein. This is going to be a very small protein, but that's what it does. Back in the 50s and 60s, when I was just a very small person, scientists discovered that there was a code in the sequence of DNA. And that code tells you which amino acid goes in which space. And it's actually a pretty simple code. It's based upon 3 letters, or 3 nucleotides. So every 3 nucleotides in DNA codes for a single amino acid. So if you want to know what the sequence of this protein was, first thing you'd want to do is you'd want to break it up into 3-letter words. So we have UUU, that would mean one amino acid. GUC represents another, AAA another, and UGA theoretically can continue. So Mike, you have to remember UUU when the time comes.
Okay.
GUC.
GUC and Mark AAA. Too late?
Hey?
Why do you think I picked? Or UGA. I'll do UG. So, the point is there a code to this?
Yes.
And there is, there's a cheat sheet, and there it is. So you now have a cheat sheet, and you can read any organism's DNA and now convert it into an amino acid sequence. All I do is match it up. So what was the first one?
You.
And I have, that was easy to remember because I had a circle, and it codes for the amino acid, whose abbreviation is PHE. Remember I said scientists don't like long things, so they take the word phenylalanine and abbreviate it to PHE. In a few minutes you'll see they're going to abbreviate it just down to P, because they get really lazy. So the first amino acid, wherever you have an EU, the cells know to put in phenylalanine. The second one was?
I forgot already. GAC?
Well,
I think it was GAC. GUC. Thank
Let's-
you.
let's
My
see.
brain is very old.
Oh, it was GUC.
GUC.
And it counts for valine. That's another amino acid. The third one was AAA, I won't even go there. Codes for lysine. The last one, UGA, says stop. By the way, stop is not a three-letter abbreviation for amino acid. It literally means the end.
Yeah.
That's how you know where it ends. So if we go back, whenever the cell sees this and is trying to read it, it knows to put in that set of amino acids. Now, the only difference between this and, let's say, another protein is size. Usually proteins can be hundreds, if not thousands, of amino acids long, so the gene for them are hundreds, if not thousands, of nucleotides long. But it's pretty simple. And one of the most amazing things is that it doesn't matter what organism you're in, they all use the same cheat sheet. So you can take a human gene and put it into a bacterium, and if you can get it to do the right things, it can read the code, because it's the same code, even though they never make that protein in real life. And that's the reason we can, by the way, have animal models that we'll talk about later on, because the code is universal. Now, very quickly, this is really just for terminology. If you want to make a protein, that means you have to first have amino acids. You get them because you eat the meat and the tofu. And two, you need the instructions. And the instructions are where?
The
In the DNA, which is in, well, we'll talk about where that is. So if you want to make a protein, it turns out it's a two-step process. The first process is you take the DNA, you find the gene you're interested in, and you make a copy of it. The copy is close to being identical, but it's a slightly different chemical. Instead, they misspell. Instead of calling it DNA, they call it RNA. There's a reason why you make this copying, so we'll just not worry too much about it, but you have to make a copy. So the first thing you have to do is you have to make RNA. And what would you think you would call such a process? I would call it RNA synthesis. But of course, if I asked you what's synthesized during RNA synthesis, you'd probably tell me, no, RNA. That's why it's called RNA synthesis. So in other words, most non-scientists would know what RNA synthesis is, so that's no good as a word, so you have to invent a new word. And so here they use a metaphor. What they say is they're transcribing instead of copying. And so they call the process by which you make RNA from DNA, transcription. Because you're basically copying, so it's a fancy word for copy. Then you take the RNA and you then use it, that's where you actually read the code to make the protein. So you could call the making of a protein synthesis. Now let's see if you get it this time. So what do you make when you're in, what do you synthesize in protein synthesis?
Protein.
Proteins. Do you call it that? Of course not, because everybody would know what it meant. So instead you call it something else. And the metaphor in this case is that you're translating it into the world of proteins, the language of proteins. It's called translation. All you really have to remember from this, and you probably don't even have to remember it, is that anytime you're transcribing, at least when you hear it today, because this isn't quite true, you are in the process of making a protein ultimately.
Question.
Uh-huh.
Yesterday, Lenore showed a slide of the blueprint, the architect or the builder. So that relates to that.
It's the exact same thing.
The DNA
So
is
the original documents, I forget the exact word, is the DNA. The blueprints, which are copied from the originals, are the RNA. Then when you make the building, you're translating that two-dimensional thing into a three-dimensional. And it is localized in different parts of the cell. for our purposes, we only have to worry about two parts of the cell. We have to worry about the nucleus, which most of us probably remember at some point. It's either that or it's in the middle of an atom. Or the cytoplasm, which is everything that's not the nucleus. DNA is housed in the nucleus and can never leave. It's too big. The structures in the, that make proteins are called ribosomes. You'll probably never hear that again. I'll already tell you ahead of time, it's not on the test. But they are located only in the cytoplasm. So since the DNA has the information, but the machines that make the proteins are in the cytoplasm, and neither of them can go to each other, and you have to have a messenger in between, and that's the role of RNA. So it's called messenger RNA when you make it. So the process is, again, you transcribe the DNA, you into RNA. The RNA then goes and is made, and then the protein folds and becomes the active protein. So, all I want you to remember is when you hear transcription, you know in the end you're making a protein. That's part of the process. So, once again, because we are having a quiz afterwards, the specific sequence of every protein is coded in DNA. Proteins are made in a step process, transcription and translation. The specific piece of DNA that codes for specific proteins is a gene. Again, this is probably in some manners can be wrong, but it's good enough for us. And this is what you inherited from your parents. When you inherit genes, you're inheriting instructions how to make proteins. And DNA is composed of nucleotides. The sequence of nucleotides determines the amino acid sequence. And by the way, your cells are not smart. If you were to be given, we have that. If the sequence of nucleotides DNA is altered, known as a mutation, the protein amino acid sequence is altered because the cell is stupid. It doesn't know, they didn't mean to put it here. They can only read what you're given. So it's kind of like going to Canada and trying to look up the word color. And COLOR, but up there, they have a different dictionary when you look it up. Probably in Australia as well. They have. So one of us may have the right dictionary, but they have the wrong, but the point is it's different. So one of our dictionaries mutated. We figured it was good to get away from the English early, but the rest of it took time. So basically what happens if you build a protein, and it's a mutated protein. You didn't know this song dealt with protein synthesis, did you? The protein isn't going to work. And that's what happens at KD. We have a mutation in a gene that codes for a specific protein. We have a change in the DNA, which means we're going to put in the wrong amino acids, and we're going to get a protein that doesn't have the right sequence, and it doesn't work as well. So far, so good. What protein is it? You all know because you've heard it time and time, I'm sure. The androgen receptor. You say, what does the androgen receptor do? Well, it binds to testosterone. And then causes whatever you hear of, if you hear that testosterone causes muscle building properties, it's because it binds the androgen receptor. And the androgen receptor and testosterone together cause your muscles to get bigger. Or it causes your voice to get deeper. It's because of the androgen receptor and testosterone working together. They're a team. They have to work together to have an effect. If you don't have testosterone, you still may have the androgen receptor, but the androgen receptor does nothing. We can call them women. Kind of a joke. That's not a bad thing.
No.
Would you rather be like Arnold? So, since it's a protein, it has a specific structure, a specific sequence of amino acids, and here it is. It's about 950 to 1,000 or so amino acids long. This is why we use only one-letter abbreviations, because it's so long. This will be on the test, so make sure you memorize it. Now, let's imagine, it's a little bit bigger, let's imagine it had 1,000 amino acids. Since the gene that must code for it must therefore have at least 3,000 nucleotides, right? Because every amino acid is caused by 3. Just say yes even if you don't mean it. Well, here's the sequence. It's about 3,000 nucleotides. And hopefully, I've never done it, if you went through them, you get the same sequence I had before. I'm not going to keep it up long enough to see if that's true. Now, what sort of mutations can you have? Remember, mutations are when you mess up the sequence of the nucleotides. Now, when I used to teach, I used a metaphor of a sentence representing the protein. And the words in the sentence represent the nucleotide. And remember, once again, remember the answer, by the way, is 3. How many nucleotides per each word in DNA? Right? Each word is one amino acid. So let's do the same thing. Let's try to read this sentence with only 2 rules. Every word is 3 letters long, and the end of the sentence is marked by an N. That would be an stop coma. So could you read that?
Yes.
Yes. And who ate the rat?
The cat.
Now let's have a mutation where we change one of the letters. Now imagine you hadn't seen the first one. Who ate the rat?
The cat.
The cat. You'd think, probably German, right? They use K's instead of C's. That's how I would look at it. Because most would say K's and C's, at least, they sound alike. So even though we had a different sequence, it really didn't change the meaning at all. Sometimes when you mutate a piece of DNA, that may have no effect on the protein. In the same way that this doesn't have an effect. How about this one?
You could probably still get that.
You might, especially if you take Latin or Spanish or French, because you know Q's in the Romance language have kind of a K sound. But it's not as good. Some people won't get that. The people who take Finnish may not get that. Because they may not realize Q's. I don't know what Finland's Q's are like. We'll assume it's not K. So this, for some people, works okay. So it works okay, but certainly not as good as either of the other two. How about this one? Would you ever know that the cat ate the rat if you had this? No. It changes the meaning completely. And so this would be a protein that essentially, if you made it with even that one change, doesn't work the way it's supposed to. And most of the time it doesn't work at all, but that's not always the case. So these sorts of mutations are called substitutions. You can also have deletions. You can actually cut out a letter. I mean, remember the rule. The rule isn't, look and see what words fit. The word is you have to read every 3 letters. So try to read this. The, ata, tat, her, ate, and it will continue. What did you get?
Nothing.
Well, I would say gibberish, but nothing is good. This is a terrible, it doesn't even make a protein. Same thing if you have an insertion. You put in one by some mistake. You mess up the reading frames, you get basically nothing. So now you're wondering, what's the mutation in the androgen receptor gene that causes KD? Is it a substitution? Is it a deletion? Is it an insertion? And the answer is none. It's a stutter. The cat. So notice you can still read the sentence, you just look silly doing it. And this was a really unique mutation. First discovered with the Kennedy gene, first for instance. Back in 1991 from Alice Spada and Kurt Fishback and other people from the lab. Now, what does it really look like? Obviously it's not like, well, here is the DNA sequence that we showed you before. And if you look very carefully, and I'll help you, it's in red, there's a sequence, to make it bigger, a sequence where it says CAG, 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 it's a repeat. We call this a CAG repeat. Again, in grad school we call that. So we know what it is. Now, in this particular case, there are 20 repeats. If we went into the androgen receptor gene of everybody in this room, we would find that, for the most part, it's different in different people. Now, because it's X-linked, men only have 1 copy, so whatever that 1 copy is that we have. For women, you have 2 copies, and both of them can have different numbers of repeats. So the number could be 15, 17, 20, 25, 30, and for the most part, so in other words, what that means is I'm about to tell a lie. It doesn't probably really matter amongst those. There are some subtle differences. If you have a higher number in your male, you apparently have less chance of getting prostate cancer. But for the most part, you never would have known that there's anything different about you if you had anything like that. The only time you see a difference is that repeat rate gets up above 40. And once it gets above 40, now you have Kennedy's disease. Right.
Did you say that a woman has two and they could be different repeats?
Yes, because they got them from different, they get them from two different people. I mean, that's why if you're a carrier, you only have a 50-50 chance.
Okay, okay.
And again, and isn't that it really doesn't matter at all, just that for us, it doesn't really matter. Unless you get above 40. Why 40? We don't know, so we're not going to. But what you might say is, wait a minute. If this is coding for an amino acid, what amino acid does CAG code for? And it is the abbreviation GLN. And that stands for glutamine. So now you have a best friend amino acid. Now, glutamine, remember we talked, I think, I said yesterday, when we go to the one-letter abbreviation, G was already used, so they took the next logical letter, Q. So whenever we see Q, that's a glutamine. So if we go to the amino acid sequence, we should find a series of 20, since we had 20 CGs, 20 Qs, and there they are. So this is sometimes also called a poly-Q disease, poly meaning many, or a CAG repeat disease. So there's lots of different ways of describing it. But that's our mutation. And whenever you have the protein with over 40 Qs in that row, that's when you begin to have problems and the cells apparently die. Since then, at least 8, and it might be 9 other diseases were found to have CAG repeats and are poly-Q diseases. They affect different genes, not the engine receptor.
How many diseases?
I think there's 8 or 9. 10, is it 9 altogether or 10 altogether?
Maybe there is a 10.
Yeah, that's what I thought. There used to be 9 and now they're saying, well, there might be 10. They all affect neurons. But because they are in different proteins, it turns out they affect different neurons, so they have different symptoms. So Huntington's disease, which is by far the most famous, affects certain cells in certain places. I probably should know, it really doesn't matter. But it's a little bit worse than ours because when it affects the motor movements, it's much more drastic and quick once it starts happening. And it also affects cognition. So you basically lose everything, and in a matter of about 15 or 20 years, and the last 5 aren't very kind, you end up dying because of it. We, as much as it's hard to say this, are the lucky ones. We have a disease that for the most part is not fatal directly. And it's very slow progressive, but it has the same mutation as these other ones. So in a sense that's kind of nice because maybe the reason the cells die are the same in all the cells. So if they find an answer to one of the other diseases, they might find an answer for us. So, and that's kind of hope. And that's one of the reasons I think why Kennedy's disease is actually so well funded. Considering the small number of people who have it, is because it's part of the bigger family. And there are reasons that we're really lucky. We know what the protein is. In many cases, we don't know what the protein is when it does. So, now, let's talk a little bit about the androgen receptor so you have an idea of how that works. So, first of all, you know it's an androgen receptor. That means it binds androgens. Androgens are just a fancy term for testosterone and its cousin dihydrotestosterone. So basically what happens is down here this, and Jameson, what color is this?
Salmon, yeah.
Okay, so the salmon thing is the androgen receptor. Testosterone is blue. And dihydrotestosterone, which is just a very close cousin, they're very similar, is pink. And they both will bind the androgen receptor. And essentially the androgen receptor goes in the cells and starts transcription. And right now, therefore, the cell's going to start...
Copy.
No!
Making
proteins. Remember I told you, transcription, making proteins. Did I not tell you that?
you said there was going to be a test.
There was part of it, was oral. Well, you should have remembered it. So in other words, what therefore testosterone does through the androgen receptor is causes the cells to make new proteins that it probably wasn't making before. If you don't have testosterone, you don't make those proteins. For the most part. And therefore, that's the difference, essentially, between men and women. Their cells make different proteins than our cells because they don't have that much testosterone. I mean, that's a generalization, but it's good enough for us. Turns out that part has nothing to do with KD directly. But it's nice to know. When you think of the androgen receptor, my mind is really warped. I think of it as a Swiss Army knife. That usually when you see it, it folds in a certain way. But if you push the right buttons, this is a switchblade, you get lots of stuff. I always wondered, do they have Swiss Army knives in Switzerland? I didn't know it was just something they made up. I mean, Haagen-Dazs is from Baltimore. And this is kind of like how my warped mind thinks of the androgen receptor. So here's an androgen receptor. It's in the cytoplasm. This over here on the left is the nucleus. There is no testosterone, because you can't see it. Now, there has to be a site that binds. There must be a binding site for testosterone. I would call it the testosterone binding site. The androgen binding site. I would never have called it the ligand binding domain. But that's what it's called. The LBD, the ligand binding domain.
What does ligand mean?
A ligand is a fancy scientific term that means a small thing that binds to a bigger thing.
And in a
Some-
Canadian accent, it's ligand, binding.
I mean ligand A? I'm sorry, I shouldn't have done that. I shouldn't have done that. I apologize. Now, there's also someplace in there you have the 20Q, and that could be a 40Q or an 80Q, and for what we're talking about, it really doesn't matter. Okay? And so this would be basically a prepubescent male, there's no testosterone, or a female. And so what the androgen receptor does, it floats around the cytoplasm. One thing you never think about, they never teach you, at least in intro biology, is that proteins don't last very long in cells, usually. They're constantly being removed and replaced. And it's, within a week or two, most of your androgen receptors have been replaced. So you have to be able to get rid of them. And it turns out that we have no problem doing that. Women have no problem. If you have, even men that don't have testosterone don't have any problem. And basically life goes on, and the androgen receptor essentially does very little. But if you have testosterone, it will enter the cell, bind to the ligand binding domain, excuse me. And then it's like pushing the button on the switchblade. That opens and causes the proteins to fold slightly differently. And it exposes stuff. I don't know if you noticed, watch it real closely. Watch the left side of the androgen receptor. You don't know how long it took me to make this. So I'll do it in slow motion. So a part of the protein now opens what was open before. It's Swiss Army knife. And this is called the NLS. That stands for the nuclear localization signal. Remember what happens, what we told you before, what happens when antigen binds to the androgen receptor, it goes to the nucleus. You probably never thought of it, it says, well, how does it know? This, as you can see, is an arrow and says, this is where we have to go. And it's not exposed unless testosterone is present. So once testosterone binds, there's a change in the folding, and it goes
To the nucleus.
Wow.
I was up to about 3 in the morning.
Can we see that again?
Turns out, yes. Any more, or are you sick of it? What happens in the nucleus? We already know that. We know that it binds to DNA and causes you to transcribe. To make proteins, exactly. And it doesn't bind randomly, it binds to specific places to make specific proteins. Now, one of the things that's kind of strange, if you think about it, is that androgen and androgen receptors make different proteins in different types of cells. So how do they know which piece of DNA they're supposed to bind to in a liver cell as opposed to a muscle cell to make different proteins? And the reason, what the answer is, and I'm going to make a very generalization, is there's a lot more complicated than this. It has lots of helpers. And these helper proteins bind to these other places that opened up when they get inside. They're called activation sites. And I don't have 2 there, but there's others. These other proteins that come in and help are called co-activators. And each cell type has a different set of these co-activators, so they join together and bind to different genes, causing different proteins to be made. Do we care about that? Probably not. But I figured I'd say it because it sounds good. But here's a picture that someone else drew. This didn't take long to get. It took me about 3 minutes to Google and copy and paste. And it's exactly what we said, but it has those co-activators present. So, but there's no movement, so it's not quite as cool. So here's the testosterone coming in, binding to the androgen receptor, which is this yellow thing. What I never showed you is that the androgen receptor has people who help it around to stay in the right shape. They're called chaperones. You want to make sure that androgen receptor doesn't do anything it shouldn't be doing. That's exactly what happens when testosterone binds, it loses the chaperone protein, goes into the nucleus. Now you probably won't remember this, but tomorrow there's a good chance you're going to hear about other changes that occur when it comes in when they add these P's. These P's stands for phosphates. If someone says phosphates are added, you're going to say, that's right, Ed talked about that. I mean, he can't tell you much more about it other than it occurs. We're not interested in it, but it does happen. These then basically join in these other proteins, this orange protein, this pink protein, and this blue protein. Now, these are the co-activators. And remember, the actual which co-activators that are present are different in different cells. And this could be the reason why only certain cells die with KD, because it might be the co-activators are also involved in somehow causing the problem. And not all of them have the same co-activators, you have to have the right co-activators to have the cell death, etc. That's at least the hypothesis. Now, what happens at the end of this? When you're making proteins, you're making proteins, and after a while the nucleus says it's time to get rid of the AR. I think it's hours. So it breaks it apart and recycles the part. Unless you happen to have a polyQ that's over 40. Then you're going to find out, it appears we're not able to break it down very well. So remember, we only have this problem if it gets into the nucleus, and we only have it in the nucleus if there's testosterone around. And you only have this problem if you have more than 40 Qs. So we'll come back. Everybody okay with that? You gonna remember that? Yeah, you are. I'm going to skip that. One of the things they discovered very early about cells from people in the spinal cord and who had KD is that inside the nuclei of their motor neurons were these big clumps that are called inclusions. You don't see them in normal cells, people who don't have KD. You might see them in other diseases, but you don't have them normal, so it's not a normal thing. And if you look at them real close, you can see, wow, how interesting. It looks a lot like a clump. And you say, well, what's in these? Well, they're kind of hard to analyze, but they've analyzed some of them. And one of the things that's in them is part of the androgen receptor. And so what it appears is happening is that, remember, if you have over 40 repeats, somehow the cell's machinery for breaking it down gets jammed. Can't break it down all the way. These parts then float around, and they may clump together to form a clump. So people who don't have CHE repeats or polyQs over 40 don't see the clumps because you're able to degrade them just perfectly. Only us guys who have testosterone over 40 we get the clumps. It was asked yesterday, are the clumps good or bad? And the answer really doesn't matter. Because what they are a sign that you have too much debris left. It doesn't matter if they're helpful or not, because not every cell gets them. But they're an indication that you have too much of those debris, and either the debris or the clumps are not good for the cells they cause you to die. Why is it they cause you to die? We don't know. But that's one of the theories, one of the hypotheses, is that it's the buildup of this stuff. And you can tell you're building up because you're going to see the clumps. Okay, so in toss, it really doesn't matter.
Is that the nucleus of the cell we're looking at, or the entire cell?
That's the nucleus of the cell right there.
And
this is a blow-up of the nucleus.
Okay.
And this is a, yeah.
Okay. All in the
Right.
You said the buildup causes the cell to die. Do you know what that time frame is?
No. Do we? No. And we don't really know what it is. The hypothesis is that it's a smaller fragment, not the clumps. And the clumps might be good because it's taking these toxic things away. But not every cell clumps. And it could be that the ones that don't clump die, the ones that clump stay alive, that's why we see it. I mean, that's kind of the theory. But the key is, for us, is that this is very typical. It doesn't matter what the cells are. If they can't get rid of the AR, they're going to see clumps in some of the cells. And we're going to use that in the future.
And we don't know if that's the cause or the effect.
That's right. Well, we can guess.
Yeah.
Now, so if you're trying to now prevent the cell death, one way you can think about it is trying to prevent the clumping, which means preventing the making of the toxic fragment, that fragment we can't break up. And there's a couple things you might try. We're only going to look at two. I stole this from somewhere. It's a little too complicated. But there are two processes by which you can destroy proteins. The one that's normally apparently used in the nuclear cell is this thing called the proteasome. We've talked about that being like a wood chipper. Protein goes in one end, chops out. And again, Lenore talked about it a little yesterday. Turns out that you can do experiments that demonstrate that if you have a 40 or more repeat, when you try to go through the wood chipper gets clogged. So the proteasome can't work, and that's why you have a buildup of these parts, because it's all clogged up. It's like a backed up drain, everything backed up. So what and then in support of this, there are drugs you can add that activate a different pathway. This pathway has a cool name. Oops, there it is. Autophagy. Or if you want to say it funny, autophagy. And people say it both ways, but I like autophagy. And this is a little bit different. It's different than the proteasome. We're not going to go into any details. But if you are able to stimulate that process in cells that have KD, essentially, you can lower the symptoms. You can get rid of those clumps. So it appears that if you can somehow prevent the accumulation of those particles, the cells are happy. And that's one of the things people try to do, trying to prevent that from happening. Now, how do you study things? And having KD is very weird, because it's because there aren't, and that I know of,